Okay, I'm going to start. Thank you everyone for attending Science Matters today. Before we start with Cecilia, I would like to invite everyone here to participate in the PNRI Rare Genetic Disease Research. We have this happening in Seattle in February 28th of next year, and it's going to be a symposium, a scientific symposium, where we have great scientific talks of the Seattle community, panels and poster sections, and an evening reception. The registration is free. The complete agenda is already in our website. Please register, send your titles for poster, and let's all meet there. It'd be awesome. So today we are very pleased to introduce Dr. Cecilia Poli, Associate Professor and Director of the Program of Immunogenetics and Translational Immunology at the Universidad del Desarrollo in Chile. Dr. Poli received a medical degree from the Universidad de los Andes in Santiago and performed her residency in pediatrics, followed by a PhD at Baylor College of Medicine, where she studied human genetics and immunology. Dr. Poli returned to Chile in 2019 to focus her research in investigating the genetics of inborn errors of immunity and autoinflammatory disorders, for which she discovered several new causative genes. Her work has a key translational aspect as the discoveries directly drive patients' therapeutic strategies. Recently, Dr. Poli and colleagues established the Deciphered Initiative for Rare Undiagnosed Diseases in Chile. This program has the ambitious goal of developing a model for low and middle income countries to provide molecular diagnosis for patients with rare diseases. Welcome, Cecilia.
Thank you, Claudia, for this very nice introduction. And most importantly, thank you and the PNRI for inviting me to this very exciting talk. I'm thrilled to be here today. I'm going to share my slides, and if it's all sharing correctly.
Yes.
So I chose this title today because I think what's exciting about studying the immune system and genetics causes of its disruption is that the cells are really easy to access. So just a needle stick away, and there's a lot we can discover through that needle stick. So I have nothing to disclose. I love this picture of this little girl full of dirt, yet not getting sick, because the immune system is what keeps people healthy most of the time. And the girl is reacting to everything that's external to her and not being infected, and is also healthy, not reacting to things that are internal to her, and is able to play there in the mud. So primary immunodeficiencies are the inability to contend with the internal or external environment. And the discovery of primary immunodeficiencies started with Dr. Ogden Bruton, who described XLA in patients with severe infections that were later discovered to have defects in BTK, that is a molecule that is required for B cell development. So these patients had no B cells and also, therefore, no antibodies to fight infection. Later, a severe combined immunodeficiency was described in patients that had no B cells, no T cells, and could or not have NK cells. And this is a picture of Dr. William Shearer in Houston, Texas Children's, taking care of the bubble, David the bubble boy, that had severe combined immunodeficiency and could not be outside of the bubble because he would die of infection. And this is really where it all started. Dr. Robert Good described these patients as the experiments of nature in the development of modern immunology. And I'm going to try to convince you today that this hypothesis still stands, because the study of these patients really enable us to understand how the human immune system works. Today, more than 500 genes are associated to inborn errors of immunity, with more than 560 different disorders, because one thing we've learned is that there's been an exploding phenotypic expansion, not only genes, but also variant-specific phenotypes. So we can have one gene associated to one or even more different disorders. These descriptions have offered great opportunities for monoclonal antibodies development, small molecule blockers, and gene therapy, so very targeted therapies. And there's been great phenotypic expansion. So as I showed you, the first disorders were associated with immune deficiency and infectious susceptibility. But then, as we discovered more diseases, disorders causing autoinflammation or autoimmunity with or without infectious susceptibility were described. Patients with immune dysregulation, cancer susceptibility, and we also found that one gene could have one phenotype or another phenotype, depending on the specific variants that affected patients. Also, severe allergy could be explained by inborn errors of immunity. And more, we are aware that one gene does not equal one disease, and there's multiple different diseases associated to many of these genes. Also, not all of them are rare. And for example, descriptions of variants in TYK2, IRF7, and IRF9 have explained cases of tuberculosis or severe influenza, which are pretty common disorders that can also be underlined by inborn errors of immunity. So we go from what's exceedingly rare to what can be a common disorders in the population. Genetic studies really transform what we understand and what we can do for patients with inborn errors of immunity. And I bring up this old, now old, but paper, but that still stands, of a cohort of patients, mainly from Texas, who were sequencing for a clinical diagnosis of severe immune deficiency, in which using whole exome sequencing enabled a diagnosis in 40% of this cohort. Importantly, some patients had novel diseases, and other patients also had more than one gene explaining the phenotype of the patient, causing a blended phenotype of two or more genes in the encoding for molecules that participate in the immune system, accounting for one phenotype in one patient. Genetic diagnosis is also very important because it matters in terms of clinical management. And when we look at the green bars here, this is the amount of patients in whom there was a change in the diagnosis. And we also can see here in yellow as there was a change in the management of the patients after they received a genetic diagnosis. When we look at another subset of patients with inborn errors of immunity presenting as hemophagocytic lymphohistiocytosis, which is a severe form of immune dysregulation, we not only find that we can find a similar diagnostic rate for these patients, but also one important thing is that only 18% of the patients had variants in the known HLH genes, and other patients had variants in other disorders of the immune systems that are treated differently. So using an unbiased genetic sequencing approach was very important because it determined what we were going to do with the patients. Cell-specific characterization and functional studies are, as I mentioned, a blood draw away. So what we do is we do genomic sequencing and sequencing analysis, but we also use a lot the immune phenotyping to feed our analysis. And then we analyze and we go back to the immune phenotyping to understand the variants that or suspected variants that we're looking at. When we have candidate variants selected, then we go ahead and do functional validation using patient immune cells that then we can model in a cell system or an animal system. These cells can be directly expanded or derived into different cell lines, which enable relatively easy molecular studies. So we always start from a very sick patient and very well characterize them phenotypically, and using flow cytometry, we can also characterize very deeply their immune system. We use whole exome sequencing or whole genome sequencing to do a genetic characterization, and these this enables the discovery of new inborn errors of immunity, but also underlying mechanisms that can lead to specific targeted therapies. And I'm depicting here the use of a specific receptor blocker, for example, for IL-1 mediated disorders. And these therapies that are sometimes developed for these rare disorders can also be applied to other disorders that are more common, like diabetes, atherosclerosis, in which these mechanisms also play a role causing inflammation. So for me, it all started with these two children that I had the luxury really to study at Texas Children's Hospital. These two children had severe autoinflammation with cutaneous lesions that on pathologic analysis showed Sweet syndrome, lung disease, both inflammatory and infectious, and severe infections, including pneumocystis carinii pneumonia, mycobacterial infections, multiple episodes of sepsis. And sequencing analysis of these patients revealed autosomal dominant heterozygous truncating frameshift variants in POMP. And they were both de novo. So what is POMP? POMP is a chaperone for proteasome assembly, and it's very important for the incorporation of the catalytic beta subunits, both to the conventional proteasomes and also to the immunoproteasomes, in which these catalytic subunits are a little bit different and have a specific catalytic capacity to cut proteins in a certain way so that they can be presented to the immune system under the MHC class 1 presentation system. So when this protein, or when the proteasome doesn't assemble correctly, because the proteasome is charged with degrading ubiquitinated proteins, these ubiquitinated proteins accumulate. This causes ER stress, an activation of the unfolded protein response, and this in turn leads to the upregulation of type 1 interferon genes that presumably causes the autoinflammation. And in fact, these patients can be treated with inhibition of JAK, which is downstream to the type 1 interference signaling. So what we can see here is that, in fact, these truncating mutations in POM really limited the incorporation of the beta subunits into the proteasome. And this is the immunoproteasome subunits. So there was a defect in proteasome assembly. And when we looked at ER stress, we saw that ATF, BIP, and spliced XBP1, which are downstream of the unfolded protein response when there is ER stress, were upregulated both in patient cells and patient cell lines. And there is also an accumulation of these ubiquitinated proteins. When we transfected this mutant into a HEC293T cell line, we could reproduce the interferon signature, that is the upregulation of the type 1 interferon genes in this cell line, suggesting a dominant negative effect of these mutations. So it was causing an interferon response, which in immunology we call an interferonopathy, and this is a specific type of autoinflammatory disorder. But we really weren't sure if it was ER stress that was causing it. We hypothesized it was that, but we weren't really sure. And there's multiple ways that you can get an exacerbated type 1 interferon response. Usually we use JAK inhibitors to inhibit the interferon response downstream to the interferon type 1 receptor. But we know that different processes can lead to this exacerbated interferon response. And perhaps this can explain why some patients do not respond to just inhibiting the interferon response signaling itself, because these broader processes can cause inflammation through also different pathways and not only the type 1 interferon response. So we asked ourselves what precisely drives interferon type 1 signature in POMP deficiency. So because we knew there was ear stress, and this is beat in the ear lumen, and this is the unfolded response pathway that has three major proteins, PERK, IRIN1, and ATF, we thought, okay, it's this UPR response, and if we block it, we're going to block the interferon response. So we used different inhibitors, STING inhibitors to inhibit the CGAS pathway, and ER stress inhibitors to inhibit the ER stress, and looked at what happened to the interferon signature. And this is looking at the delta CT. And this is my former PhD student that did this work along with all collaborators in Germany. And we really saw nothing. To our surprise, the ER stress did not seem to be driving this interferon response. At the same time, we learned that Seth Masters, along with Sophia Davidson in Australia, were hypothesizing that it was the misfolded IL-24, that is a cytokine, got into the cytoplasm, and this was recognized by a molecule called PKR, and this was what was driving the interferon response. And they had some beautiful data from mouse models, and we collaborated with them to look at patient cells. And indeed, what we saw when we used a PKR inhibitor was that we had significant change in the delta CT for the interferon response. And we saw this in both patients. And this is clearly shown here when we look at the ISG score. So looking at different genes that participate in the interferon response. And while the ER stress inhibitors did not do anything pretty much to this interferon response, when we're using C16 to inhibit PKR, this significantly dampened the interferon response, suggesting that really it's PKR what triggers interferonopathy in this form of proteasome-associated autoinflammatory disorder. But then we had explained the autoinflammation, but we really didn't know what was the mechanism of immunodeficiency. Why are these patients getting infection? Whereas other forms of proteasome deficiency affecting other molecules of the proteasome didn't really seem to have immunodeficiency. So we also asked, is this exclusive to POMP? And if it is, why? So the proteasome is very important in the immune response. It participates in the creation of the peptides to be presented in the MHC class 1. For the immune response, it controls apoptosis and various cell cycles. It's also important in immune signaling because it degrades I-kappa B alpha to allow NF-kappa B signaling. So as I said before, patients with other forms of proteasome-associated autoinflammatory disorder that had either autosomal recessive or digenic variations in the catalytic subunits of the proteasome had neutrophilic dermatosis, had lipodystrophy, had somewhat low CD8s, but they had no severe infection, even on immune suppression with JAK inhibitors. So we suspected these patients were maybe compensating with conventional catalytic subunits to be able to create a relatively functional immunoproteasome. But our patients were really sick. And when we looked at the cytokine production in their T cells, we saw that they produced almost no interferon gamma or no TNF alpha. So they were really dysfunctional, explaining why these patients were getting these severe and disseminated infections. So we hypothesized that maybe because POMP is a chaperone for both the standard proteasome and the immunoproteasome assembly, these patients were not going to be able to compensate, creating a pan-proteasome defect that would impair NF-kappa B signaling. And therefore, because the release of interferon gamma and TNF alpha by T cells is dependent on NF-kappa B, they would have immune deficiency. So we looked at I-kappa B alpha degradation in POMP patient cells. And much to our surprise, instead of having decreased degradation of I-kappa B alpha because of a dysfunctional proteasome, we had augmented degradation of I-kappa B alpha. And when we transfected the mutants into a cell system and using a Firefly Ranilla system to look at NF-kappa B activation in the nucleus, we saw that these mutants of POMP were exacerbating the NF-kappa B response as opposed to decreasing the NF-kappa B response. So this did not seem to be the cause of immune deficiency. We inhibited the interferon response with this type 1 interferon inhibitor, and nothing happened. So this was not secondary to the interferon response. And when we used C16 to inhibit PKR, we were also able to inhibit NF-kappa B. So this suggested that PKR is stimulated by the unfolded IL-24 is not only triggering the type 1 interferon response, but also an exacerbated NF-kappa B response. And this is important because we knew from data from the NIH that have the largest cohort of patients with proteasome disorders that they don't respond well to the inhibition of JAK signaling and the type 1 interferon response. And this could be maybe explained because they also have an exacerbation NF-kappa B producing inflammation. So uncovering this underlying mechanism is important to decide what we do with patients or understand why they do or do not respond to certain specific types of inhibition. So we continue to question ourselves what was then leading to immune deficiency and severe susceptibility to infection in these patients, because patients that had mutations in other proteasome subunits didn't have this severe infectious phenotype. So we know that POMP is critical for the sequential incorporation of the beta subunits into the proteasome, but very importantly for the incorporation of beta 1I and beta 2I into the immunoproteasome. And the incorporation of the beta subunits into the proteasome is sequential and occurs in pairs. So 2 beta 1 subunits are followed by 2 beta 2 subunits and so on and so forth to conform the complete proteasome. So in 2021, specific mutations in PSMB9, this G156 mutation, was also associated to autoinflammation, interferonopathy, and severe immune deficiency. PSMB9 mutations had been described for other types of autoinflammatory disorders of the proteasome, either alone or in conjunction with mutations in PSMB8. But this, for the first time, there was this description of, again, autosomal dominant mutations causing both autoinflammation and immune deficiency in a with a dominant negative mechanism. And it turns out that this residue is crucial for the beta 1 interaction in the sequential incorporation of proteasome subunits. Similar to POMP deficiency, they observed this dominant negative effect because this interaction was disrupted. Interestingly, and similar to also POMP deficient patients, the catalytic activity of the proteasome was disrupted, but it was not severely disrupted. It is the conformation of the proteasome as a whole that was mostly impaired. And interestingly, this year, autosomal dominant mutations in PSMB10 encoding for beta 2 immunoproteasome subunit were described in 6 unrelated families to cause a severe form of combined immunodeficiency with inflammation that we call Omen syndrome. So this was interesting because now we have 3 autosomal dominant mutations causing immune deficiency and autoinflammation. Turns out that this specific residue mutated in these patients. There were 2 different residues in different families. The most common one was this GLI201, is crucial for beta 1 interactions and beta 1 beta 2 interactions. Again, disrupting the proteasome assembly in a dominant negative fashion. So with all of this led to understand that specific mutations in PN79 and PSMB10 and truncating variants in POMP lead to what we called proteasome-associated autoinflammatory disorders with immunodeficiency by a dominant negative mechanism, probably by the disruption of the sequential incorporation of catalytic immunoproteasome subunits, specifically this interaction between beta-1 and beta-2. And this probably occurs at the thymic level. And we still need to understand what is the role of the proteasome in the thymus beyond MHC class 1 presentation. To continue with the story of the POMP patients, because they were so severely immune deficient, we decided to transplant them. And this was the work of Dr. Caridad Martinez in TCH. Both patients engrafted beautifully with 100% of donor chimerism. The interference signature was significantly reduced post-transplant, as you can see here in the dashed bars. And the proportions of their T cells improved. And their cytokine production that was nearly zero, as I showed you before, pre-transplant for both patient A and patient B, was completely recovered post-transplant for both patients, suggesting that replacing the immune system of these patients and the immunoproteasome could cure the disorder, despite POMP being expressed ubiquitously in different cells. And this is the picture of one of the patients going to school several years later after transplant with no signs of autoinflammation, no infections. He's wearing no mask and going to school. So really no infectious susceptibility. What the other patient, not shown here, does have a little bit of pulmonary inflammation, and we wonder if this could be due to local proteasome dysfunction in the lungs. So take-home messages about this POMP and proteasome disorders that we learned by studying these patients is that interferonopathies are heterogeneous, and understanding what is the underlying mechanisms driving the interferon response may improve therapeutic targets. Additional immune pathways leading to autoinflammation may play a significant role in driving inflammation. So previously, we thought only type 1 interferons were driving the autoinflammation in proteasome disorders, but we learned that NF-kappa B was also causing autoinflammation, and these two could be independently targeted or explain why patients were not responding 100% to JAK inhibitors. So both type 1 interference and NF-kappa B dysregulation seem to be driven by PKR signaling in these patients. In other patients, maybe there's other mechanisms underlying this interferon response. And this proteasome beta subunit interaction seems to be critical for immune function, and the disruption of this interaction specifically is what is probably leading to immunodeficiency. So switching gears a little bit, then I went back to Chile, and very thankful to Dr. Lovsky and Jen Posey, who agreed to continue to study Chilean patients with suspected immune deficiency. So this is Chile, the long country in South America. And we have an at-mixed population. 40% of our population is native. We have a large European population of more than 50% and very little African descent. So we studied so far, because we've continued to enroll some patients and study patients that we've enrolled earlier. 53 families, 62% with immune dysregulation, and others, really several different forms of primary immunodeficiency. Most of the patients in this cohort were sequenced as trios, so the proband with both of the parents. And 23% of these cases are solved. So as you can see, lower solve rate than what we found in the Texas cohort. So there's 77% still of unsolved cases. Interestingly, within the solved cases, we found 2 novel genetic diagnoses that were published separately, CARD11 and FNIP, and all disease-causing variants in known inverters of immunity genes were novel. So this contributes to the knowledge of immunodeficiencies and to attribute pathogenicity to other variants that had not been previously described. So within this cohort, I want to tell you the story of this little girl who was 7-year-old at the time that she was referred. And she had recurrent infections and had no B cells on her immune phenotyping. She also had an intra-auricular communication that had been surgically corrected and had mild hypertrophic cardiomyopathy with recurrent infections that even led to lobectomy at age 6. On immunologic evaluation, the B cells were absent completely in the periphery. So by doing whole exome sequencing of this patient, we found homozygous splicing variant on this gene called FNIP. It had a very low frequency, a very high POI, and a CAT score of 26, so very likely to be pathogenic. This variant was inherited by uniparental disomy, which was another interesting thing. By looking at the literature, we found that there was already a mouse model of FNIP1 deficient mice that had heart defects and also no B cells. We went to GeneMatcher, and this helped us to connect with colleagues in Italy and the US that had 2 more patients with similar clinical and genetic findings. And this is the mouse model. So FNIP deficient mice had cardiomyopathy. And as you can see here, the mass of the left ventricle was much higher in FNIP deficient mice. And our patient had no FNIP expression on his T cells. What is FNIP1? Follicular interacting protein 1 is required for the recruitment of mTOR-C and TFE3 downstream to the B cell receptor. They are widely expressed in human tissues, and FNIP1 and FNIP2 interact with the C-terminal domain of folliculin. FNIP1 has a non-redundant role in the bone marrow in early B cell development, in the myocardium, and the skeletal muscle. All of these tissues express more FNIP1 than FNIP2, suggesting that FNIP1 has an important role in these tissues, and in line with what we found in the patients. FNIP1 and FNIP2 participate in both mTOR-C dependent and independent pathways, depending a little bit on the cellular context. And in the bone marrow, what we saw was in a stop at this point of maturation. So if we look at BTK deficient patients with X-linked A-gamma globulinemia, there is a stop at the pre-B cell level. So pre-B cells accumulate in the bone marrow, where here we had normal amounts of pre-B cells, but these immature B cells were accumulating in the bone marrow, and they were not able to go into the periphery. Therefore, the patients had 0 B cells. So FNIP deficiency probably hampers B cell metabolism, as patients show increased numbers of mitochondria and mitochondrial activity in the bone marrow. This was unfortunately only examined in 1 of the patients because we didn't have bone marrow available of all of the patients studied. So the mechanism of B cell deficiency, FNIP deficiency, are not yet clearly understood, but there's a role for AMPK-dependent apoptosis but deletion of AMPK or attenuation of mTOR-C does not modify the B cell deficiency. So this suggests that there are additional contributors that we do not yet understand that are underlying the B cell deficiency due to a disruption downstream to the B cell receptor. So along with Dr. Saitini in Italy, we described this 3 families. So family number 2 is the family we identified, and all of the patients had absent beta cells, hypertrophic cardiomyopathy, and neutropenia. All had synopulmonary infections. One patient had developmental disease, and one patient had Crohn's disease. Importantly, we were called by other colleagues in South America and said, well, you published this paper. We think we have a patient with a similar phenotype, and identified this 5-year-old female with A gamma, absent B cells, normal T cell counts, multiple pneumonias. At the time, she had a normal cardiac MRI, but on follow-up, developed cardiomyopathy, and I'm going to tell you why. And then another patient with a deletion that encompassed the FNIP gene that had also a similar phenotype. So we studied patient 4, also had no FNIP protein expression. And at that time, the patient also had arthritis. So she was on methotrexate and had no cardiomyopathy. Surprisingly, her arthritis got better. She came off the methotrexate. And because we had warned this family about possibility of cardiomyopathy, soon after she discontinued methotrexate, she developed cardiomyopathy. And this is interesting because we do not know what is really leading to cardiomyopathy in these patients, and what is the role of dampening the immune response in controlling the development of this kind of cardiomyopathy. So at first we thought she didn't have cardiomyopathy, but then she developed it upon suspension of methotrexate. All of the patients seemed to have neutropenia. The role of FNIP1 in neutrophils is not really well understood, but there is a participation of this molecule in autophagosomes. And so we were interested in characterizing the neutrophils of these patients. And what we found is that at least phenotypically, they were similar to control neutrophils, and that they were activated normally. So apparently, FNIP deficient patients can present with neutropenia, likely due to increased autophagy, and existing neutrophils seem to function normally, at least from the initial experiments we've performed. But this is something we're actively studying with our collaborators in Italy. So more soon on what is the FNIP role in the immune system. But what happened to our unsolved cases? As I mentioned before, 77% of our cohort was unsolved by whole exome sequencing. But we did look very thoroughly at the variants that patient had, and we described some strong candidate variants in genes that were related to PIDD genes or PIDD pathways, genes that were expressed in the affected cell types, and with strong literature supporting or preliminary functional studies supporting a possible pathogenicity. So we're studying some of those now. And then we also find some likely candidate variants, so in genes that are related with PRDD or known PIDD pathway, but we really didn't have strong literature or preliminary experiments to support pathogenicity. So these are 4 patients under study for strong candidate variants now. And so we hope to update you on this sometime in the future. Again, thanks to the generosity of our colleagues in Baylor, we were able to establish this initiative along with Dr. Gabriela Repeto, a geneticist in Chile, called Deciphered. It means Decoding Complex Inherited Phenotypes on Rare Disorders. So extending a little bit out of the immune system and into patients with other types of rare disorders. So again, Dr. Lupsky and Jen were generous enough to help us start this process in sequencing all of the first patients in this cohort, and also helping us train our team for genetic analyses of these patients. So really launching our capacities to do this ourselves in the subsequent years. So the aims of this initiative are to implement a hybrid model of genomic analysis for patients with undiagnosed diseases. It turns out that sequencing in Chile is still very expensive. So first we sequenced at Baylor, then we sequenced in China, but because Dr. Lobsky, Jen, and their teams helped us really train our team in the analyses for gene discovery. We were able to then analyze these results locally. We recruited patients with multiple congenital anomalies, neurodevelopmental disorders, and immune dysfunction, and assessed them both phenotypically and genetically. We also wanted to evaluate the personal, clinical, and health system effects of having a genetic diagnosis in these families, and ongoing studies of interviews to these patients and their families to understand what is the impact of sequencing analyses for these families are ongoing now. So there was first a collaboration phase, and then we performed exome sequencing abroad, but all of the rest was done locally by a multidisciplinary team. We recently published the results for the first 103 probants, including 246 participants, considering the relatives of these patients. And we have a diagnostic rate 41%, in which these exome analyses revealed were informative. And we found unique findings. So 50% of the causative variants that we identified were not previously reported. They were novel. So we uploaded them to ClinVar to make these available to the community in general. Secondary findings were also identified in 8 to 9% of the patients. And to continue with our collaborations, we're now working in a cohort of patients with a subset of inborn errors of immunity characterized by autoinflammation. So what we're doing with these patients is a little bit different, because we want to do genetic diagnoses, but we also want to characterize the different inflammatory pathways using RNA-seq and some established assays to assess the interferon pathways, STAT phosphorylation, caspase activation, and also NF-kappa B. And of course, perform candidate validation in both in cell lines and model organisms. And we've called this initiative GenAid. So these are the very preliminary results. We have 104. It's closer to 120, but the last time we looked at it was 104 patients. We used patients with a known genetic diagnosis to use them as positive control for biomarker identification, and then focused on genetic diagnosis of this 72% patients presenting with autoinflammatory disease, yet not a known genetic cause yet. So these are our positive controls. There are 18 patients with mutations in the different pathways that are known to lead to autoinflammation. So inflammasomopathies affecting the inflammasome, interferonopathies are those known that disrupt the type 1 interferons, relopathies disrupting NF-kappa B, and immune dysregulation. And we're looking at the transcriptomics of these patients to then be able to compare with these patients in whom we do not know a genetic cause, and hope to have this transcriptome analysis help us also interpret the genomic analyses, and let one feed the other for discovery and also biomarker identification. So far, there's 23 of these families sequenced. One of them, we have a candidate gene who is undergoing functional analysis. We have another 3 families with candidate genes that we hope to move to functional analysis. So it's been very interesting living in even in a relatively small cohort to be able to find new mechanisms and new genetic disorders. So in conclusion, inborn errors of immunity offer the unique opportunity to study how the human immune system works and can be modulated. Functional studies are facilitated by the availability of cells a needle stick away, and this accelerates discoveries and offers opportunity to this bidirectional bet.
Cecilia, you are muted for some reason.
Are you still able to hear me?
Yes, now we are.
Because as you could see here for POMP and also for FNIP, what we did both with genetic discovery and the immunophenotyping really had an impact to what we did with the patients in terms of management. Studying Chilean patients with an admixed genetic background is relevant for discovery. We discovered novel variants, novel genes causing inborn errors of immunity, as well as other phenotypes outside of the immune system in our Deciphered initiative. Whole exome sequencing and team-based analysis is a powerful tool for diagnosis and discovery, even in small countries with small cohorts, as we could see in our experience with the Deciphered initiative. Collaboration across the globe is really crucial for discovery, and I'm extremely thankful to all of our collaborators, especially to Dr. Lipsky and Dr. Jane Posey. So I want to leave you with this. It is the long history of humankind and animal kind too, that those who learn to collaborate and improvise most effectively have prevailed. It's by Charles Darwin. So with collaboration is not only fruitful, but also fun. And this is Dr. Posey when she came down to Chile, other members of the Baylor team, Dr. Lipsky, Jordan Orange, lots of fun. So with this, I come to the most important slide of my presentation. I'm very thankful for the patients and families that have participated in all of these studies, to Dr. Jordan Orange for really getting me started in this field, to Dr. Lipsky, Dr. Posey, Ivan Chin, and the BCM Breger Consortium for all what they've enabled in terms of discoveries with our patients, all of the immunogenetics team at Baylor, our collaborators in Australia, Germany. The FNIP patients turn to be all from Argentina. So this is Dr. Nieves in Argentina who discovered these patients. Dr. Gabriela Repeto leading the Deciphered team and investigation for rare disorders in Chile, my clinical team at Hospital Roberto del Rio, and the clinical team in Chile that always refer patients to us to be studied, my all-girls team in the lab, university, and funding sources. So thank you all for your attention. And I leave you with a picture of Santiago with the Andes behind, and I'm open to questions.
Thank you, Cecilia. Very nice presentation. Let me see if there is anyone that wants to unmute and ask a question, or in the chat. Meanwhile, I have a few questions. So one of them is the fact that the solved cases in Chile, you have like almost, I don't know, 12%, 15% less than the one you did in Baylor. Do you think this is because of the cohort itself, the type of diseases that has less genes that are known, or do you think it's because the population, which I mean could be a combination of both, but I wonder if you have a thought about it.
I think it's a combination of both. Probably at the beginning, when we started enrolling patients, we didn't enroll the severest phenotypes. And I think we analyzed this for the Deciphered cohort, and we didn't find clear correlation between the severity of the phenotype and the diagnosis. But I think for immune disorders, if there's a severe phenotype, the likelihood that you find something is higher. And at the beginning, when we started enrolling patients, we were less strict in our criteria to enroll patients. So I think so that is one reason that it could explain. The other is that our population is different and we could have new diseases. And as I mentioned, we do have candidates to validate. So those will be interesting in the future. And at least two of them, we really have nice preliminary data that makes us think that we're going in the right direction. Unfortunately, although it's easy to access the immune cells, really proving this sometimes is difficult if you don't have more patients with mutations in the same gene in different families. And we put these in Gene Matcher. We found matches for ethnic, but we haven't found for the other genes. So we're awaiting on that, hoping that this could help really solve these cases.
Mm-hmm. Okay. I'm also wondering if, because I mean, what I love about primary immunodeficiencies, and we have some of your patients here with us, right? So is that the fact that once you find the mutation, you can do a functional study that's going to be meaningful, right? Most of the time, if you are looking into neurodevelopmental diseases, if we do blood, it's not helpful. I mean, it can be a little helpful, but it's not going to be completely resolving. In the case of primary immunodeficiency, it's really, really helpful because that's the tissue that we are most of the time, right? The other thing is that there is a translation impact that is immediate. You can do something for a lot of these patients. That is really like rewarding, right? So I don't know. I wanted to see if you have any comments about that or.
Yeah, I think what you're saying is exactly right. I am totally, in love with these disorders and with the ability to study them genetically and functionally, because as you said, it's really rewarding. I don't know what I would do with my patients if I didn't have a lab anymore. Like I would feel totally helpless because I know and have experienced that if you are able to study them and discover something, or even not discover the genetic cause, but even discover what pathway is disrupted, what cell is disrupted, you're able to do something for the patients. And that's really rewarding. And also the fact that we can transplant the genetic and cells and change the defective immune system for a new one is also really nice because we can cure these patients with transplant. Of course, it's not easy in all of the patients, and it is fairly challenging, but it is something that can be done as opposed to other disorders. It's important to have functional analysis because when we first encountered the pump patients, we thought, well, this is a molecule that is expressed everywhere, so we're not going to fix the patients by transplanting them. But what this experience shows is that despite the ubiquitous expression of the protein, we're able to almost cure these patients completely by replacing their immune systems for a healthy one. And what really helped in these two cases was when we looked at the T cells and said, well, they're not working. This is explaining the infections. We thought that they would not do great and that inflammation would continue, but it seems like if you fix the immunoproteasomes, patients can do fine. Of course, we need to look at them in the future and see if there's other manifestations of local inflammation.
Yeah, that's very nice. And the other thing is that the fact that you're finding these genes and also can help with more common diseases. You just mentioned about the arthritis, right? And allergies as well. Yes, I really, really like the vitamin deficiency. It's really nice. Thank
Yeah,
you.
cool. And I think we're more and more, when we thought about inborn errors of immunity, we were thinking on rare, okay? This needs to be rare. But more and more, I'm sequencing the common patients with just, a severe phenotype of common allergic dermatitis. Because although it is unlikely that you find a monogenic cause, if you find it, it is totally a game changer because you have a target molecule. So I think we're going to be sequencing more of these patients and understanding the breadth of each of these diseases, because now we think it's all severe and rare, but maybe there's milder cases out there that can also be explained by a monogenic cause.
Amy, do you want to ask a question?
Yeah, I wanted to kind of touch a little bit on this kind of idea about, how, like with POMP, right? This is like and I don't know a lot about it, but this is kind of a general process, right? You're talking about chaperones and you're talking about, the unfolded protein response. And so, and you guys and your collaborators, effectively got to identify in that whole sea of things that is changing, apparently the actual molecule that needed to be was the source of some of this. But is there some kind of general way, like are there particular omic technologies or other data types that can be integrated to try and narrow in, right? Because we haven't studied it, but we've focused on the unfolded protein response because we've studied aneuploidy in the past, and aneuploidy and triggers the unfolded protein response, but it's not clear how directly related any of that is to some of the phenotypes, right? So is there some kind of clever and more general way that you're thinking about to try and narrow in on kind of how to get to kind of a proximal disease phenotypes and what is relevant in these general effects?
Oh, this is a very interesting question and a very difficult one, because the ER stress and it's not easy to assess, and the proteosomes, it's a macromolecular complex that molecules can compensate, and they also have tissue-specific roles that we don't understand yet very well. So for the functional evaluation of the proteasome, what has been used, there's a different way. You can look at the proteasome assembly using these native gels and seeing how they incorporate into the proteasome. And you can also look at the catalytic activity in several ways. You can look at the, how the substrates are degraded in enzymatic assays. There's also other luminic assays. One is called proteasome glow, that allows you to assess the catalytic activity. But these are two activities of the proteasome that we know are important and we're assessing. We can also look at the accumulation of ubiquitinating proteins. But there might be other roles that we don't understand very well, and therefore we don't know how to assess them really well. So,
I guess along the lines of, this IL, and I forget which one it was, but, being kind of one of the really important factors, the unfolding of that one, like, yes, you can look at, the overall activity of the proteasome and things like this, but like, is there some kind of clever way to narrow in on, like, is it everything, or is it just really one factor, right? This was kind of something that, when people talk about like synthetic lethality, right? Is it just that there could just be one essential protein, or it could be that you're screwing up everything and the cells die, right?
No, I don't know the answer to this question. It's a very we've been thinking about it a lot. And then we thought that, the ER stress was easy to assess, but it turns out that a lot of these inhibitors are not specific. So they inhibit a lot of things, and it makes it difficult to draw conclusions. There's mouse models of, the different molecules in the ER stress to assess each one. But none of those are perfect, and that I don't know of a very good readout that would allow us on its own to narrow it down. I think it's cell specific, it's protein specific, and of course proteomic. I would love to have proteomic analysis of all of these patients and see, what proteins are being broken down. If these are not being broken down, then maybe this is the defect. But I don't yet know anybody that has done this, because I would assume you need a larger cohort of patients with each specific defect. And as I showed for the PSMB9 G156 mutation, so it's also mutation specific. So what one specific mutation does is different from other mutation in the same gene does, and how it affects the catalytic activity and the immunoproteasome assembly. So no, I don't know of any like easy, straightforward ways. But thank you for that question. We've been banging our heads on that one.
Okay. Any more questions? Oh, Michael.
Yeah, I had a quick question. This is really exciting and a lot of really cool work. And one of the interesting things that you could get those transplants to work so well with such a big clinical success. I was wondering, how close do you think things are to being able to do sort of gene therapy targeted to each of these different monogenic mutations you've found? It seems like it's really promising to be able to sort of take the cells out, do the homologous recombination or something to repair the cells and put them back in. If a transplant will work, then an auto transplant of gene mutated cells.
That's an excellent point. So gene therapy has been developed for several immune deficiencies. So for ADA1 deficiency causing SCID and CGD, and now FOXP3 mutations causing IPEX syndrome, but there's no initiative that I know of for these disorders. There's no reason why it couldn't be done. I think it's just a matter of the N of patients. So I think there's overall, at least described in the literature, less than 20 patients with proteasome disorders described. So it's a very expensive thing to develop for very few patients. And I think where it's all going there is to try and fix these disorders that affect wider population, but there's no reason why this could not be done as it has been done for other immune disorders.
That's really interesting. Yeah, thank you.
Thank you.
Okay. Anything else? Okay, I think we are on top of the hour. Thank you so much, Cecilia, once more. And I just wanted to mention that next month in January, we'll have Dr. Kathleen Burns from the Dana-Farber Cancer Institute talking to us. And yeah, everybody have a great holiday. Thank you, Cecilia, again.
Thank you, Claudia, and thank you everyone for listening. Bye.
Thank you.